Biodiversity matters, and that's because it builds the resilience and the balance of an ecosystem.
Biodiversity refers to the whole variance of life we have on Earth. We have some evidence now that the way the human health developed through life, it's linked to the quality of microbes that you get exposed.
Biodiversity is already decreasing every year. It's a big problem for us, for the humans.
We have very strong evidence that if you grow in an environment that you're exposed to, like soil microbes with fresh air microbes, the chances that you develop allergy or asthma or any chronic or inflammatory disease is way lower.
Inside a city, green spaces are limited and there is only very few habitat, and that's why it's also important to look at it directly in our neighborhoods.
In a city, trying to improve and promote biodiversity will protect us from extreme events. So extreme temperatures, extreme disease events, or like any new epidemic.
More than half of the world's population lives in cities by now. That's why it's important to also look out for the health of those people that live in huge urban areas.
GEBIKI stands for Linking Genomic and Remote Sensing through AI for Efficient Assessment of Biodiversity.
The GEBIKI project is a collaboration of remote sensing researchers like me from TUM and microbiologists from Helmholtz. And together we want to see how the urban structures have an effect on biodiversity, especially looking at parks in Germany. The biologists take soil samples and water samples in the park, and then they take those to their lab, analyze them, and see how the biodiversity is in all these samples. And on the other side, I'm working with satellite data to actually provide information on how the city looks like from a broader perspective in the surroundings of the park.
We're going to sample the soil, right? So basically we need to hammer, and we have two samples here, so we have like 0 to 10 and 10 to 20 centimeters deep, because then we also see how the effects change in the layers of the soil. And then we sample across the plot. So in total we have 80 samples.
Okay.
That's it.
When we go for the sampling here in Munich on the English Garden, we want to use this park as a test ground. Like the microbes, they are very sensitive to very small changes, right? So they change like in a matter of centimeters, but then we wanted to see how those changes behave in different scales. We have an anchor plot, which is the one where we start, and then we need to measure like a distance from this plot. So we are working with like 10 centimeters, 1 meter, 10 meter, and 100 meters.
I hope I can cross here. The next plot is across the river, so I need to find a way if I can cross.
It's a very ambitious project. That's what fascinates me the most. So the ideal scenario would be exactly to know with how much confidence can we predict biodiversity responses with satellite data.
So now we need to finish the marking of the 100 distance plot. We need to come up with like a solution. Everything comes from the research question first, and then you need to see what you need to do to achieve the answer.
How was your...
It was fine. It's very cold, but now I feel refreshed. Yeah. So now what we need to do, we're going to start sampling the soil cores.
My part on the project is about the microbiology. We take the soil samples, and then I did the DNA extractions and bioinformatic analysis as well.
Yeah, it's a lot of work like for the sampling. So we bring those samples back to the lab, and then we perform the DNA extraction of each sample, and then we need to sequence each sample, and then we need to do the whole bioinformatic processing, which is basically the process of getting the DNA strain, giving a name to it, giving a function to it. So for each sample, and then we need to put this together and try to make sense afterwards. So it's quite a lot of work. Yeah.
Biodiversity is very important because it's the support of the life on Earth for everything.
We have for sure much more like microbial cells living in 1 gram of soil than people in the whole planet. This microbial community responds to the change in the environment. So if you have like this place here where you have a lot of people like drinking, smoking, and then you have also the cars around. So this gets imprinted in the soil, and then we can see how the microbes react to this. So we are using them like as bioindicators now. So we have this design where we have like samples taken in different plots along the park. We go to the next plot. So it's far away, close to the Allianz Arena at the end of the park. And then there it looks completely different. We chose the locations for the sampling based on the assessment of local climate zones. We want to see how those changes in the usage of the park influence the microbes in the soil.
I'm working on the local climate zone classification. We do that with the satellite images. When you zoom in, you can see more green parts. This is classified as a park. Blue parts are classified as lakes. And the dark areas are more dense populations. The brighter orange ones is more open settlements. And that data was then provided to the microbiologists so they can informatively choose their sampling spots. This classification is then supposed to be a proxy for how big the human impact is on a certain area, because we want to see how this human impact then has an effect on the biodiversity. AI in this project is used from my side as an analysis tool. We're using a model that's capable to process and analyze huge amounts of data. And towards the end of the project, we want to collect the data that we have over all this time, over the course of the whole project. And I also have time series data from the satellites. The next step is to build a new model. So we want to kind of overlay both data to try and see if there is a correlation, so a connection between the changes of biodiversity over time and the changes of a city during that time. So we can see changes that have happened over the last 10 years.
I think it was here, right? Oh. And now we need to cross to the other side, and then that should be fine. I think it doesn't get any better than this. Maybe we mark the anchor point we do here, and then we measure up and here, this way. So we do it like this.
We actually have a lot of scientists that works on biodiversity monitoring, but at this scale is the first time.
Yeah, I think it's fine, it should be fine there. It's okay.
The project, it has a time frame now of 3 years. We are like getting closer to the end of the first year. So in the first stage, we are mostly studying the dimensionality. We are going next year, in 2026, to a few cities here in Germany to try to see how this, the local climate zones, how the AI prediction from the remote sensing really applies to real-world environment.
I think this is the most chill.
Yeah! Well done, guys. Good work. I'm satisfied, we're happy and excited for the results also. Let's see how it will be.